Two graphs that we want to look at. One is called the absorption spectra, and one is called the action spectrum, because in this case there's only one thing we're following, so spectrum singular and spectra plural when we're looking at multiple pigments. So let's take a look first at the absorption spectrum, or spectra, of these different pigments. First thing that you'll notice is, of course, chlorophyll A and B closely overlap each other. Right? And beta carotene overlaps to a degree, but is also distinct in that it doesn't have this what do you call it? There's a term for having two peaks, bimodal peaks, maybe. But in any case, it only has the one peak. And so what we're looking at here is the wavelengths of light where these different pigments absorb. And so beta carotene absorbs from about 375 nanometers over to about 525 nanometers. All right? And if we forget about the pigments for a moment and just ask, what are those wavelengths of light? If we were to look at wavelengths of light that were 400 nanometers, we would sense violet, 450 blue, 500 green, 600 yellow, 650 red. All right? And in between, we'd see some other shades like orange, for example, or aqua, or I don't know what you would call between blue and violet, lavender maybe. But we can see these different wavelengths of light and perceive different colors, right? And that's actually because our eyes have pigments that are absorbing light and transmitting signals to our brain. Okay, but for plants, they're absorbing light, and if they're absorbing light in a certain wavelength, that means everything else gets reflected. So for beta carotene, it absorbs the violet, blue, green wavelengths, but it has zero absorption in the yellow and red wavelengths. Okay, so that means when visible light hits a beta carotene molecule, part of it gets absorbed by the molecule and we don't see it again. Part of it gets reflected. The part that's reflected will be the light in the yellow and red wavelengths, which is why the baby's orange, right? That's why we perceive beta carotene as orange. And in the case of the chlorophyll, it's a little more complicated. Chlorophyll, both A and B, I'll just speak of them kind of collectively, absorb in the violet and blue, and then they have this sharp decline that crashes right before we get to green. And then there's almost no absorption until you get back to red, and then we have this second little peak. So chlorophylls are nice pigments because they grab a lot of the visible light spectrum and harness that energy. Okay, carotene is a little less productive because it only harvested one region of the visible light spectrum. But what this means for the chlorophylls is that it's absorbing violet and blue, it's reflecting green and yellow, and then it's absorbing red. So if we perceive a plant like our jade visitor here, who is green, the absorption of the visible light that is hitting that plant sucks up the violet and the blue and the red, and she's using that to make, to produce oxygen and carbohydrates, but she's not absorbing the green and the yellow wavelengths, and so that's reflected back to our eyes. All right, this is true of anything that we see color for. It's not just about plants, plant pigments. And so the translation of that then for the absorption spectrum is the amount of absorption as a function of the wavelength of light. In the case of action spectrum, now we're asking how much photosynthesis is occurring at these different wavelengths. Not how much pigment absorption happens, but how does that translate to actual photosynthesis? And you can measure photosynthesis in different ways. This is a very generic unit relative rate of photosynthesis. But you can measure photosynthesis by putting a plant in an enclosed structure like a sealed box, and then you can measure the change in oxygen and carbon dioxide that happened inside that closed chamber. So you can actually measure specifically how much O2 is released, how much CO2 is absorbed, and by doing that you can measure how much photosynthesis is happening. So this is it's a chemical reaction that has a measurable outcome. In the case of our action spectrum here, if we have those three, actually I think I've got them both on one slide. Do I? I do. In the case of these three pigments, all of them are pulling in light in this range of the graph, and then the chlorophylls are pulling in light here, and so we expect the amount of photosynthesis to be highest where the multiple pigments are absorbing, and we expect to be lowest where they're not absorbing very much, and then we expect it to go up again where there's more absorption. So anywhere the pigments absorb, photosynthesis is going to happen, right? And so that's the difference between our action and our absorption. We could do absorption for anything, right? We could put, we could put pigments from vertebrates, animals in there. We could put anything we want in there and ask how much light is absorbing. But the only thing that's going to do photosynthesis will be pigments in plants. Okay, how about that? Isn't it beautiful? I think so. She thinks so. That's you. You're doing that right now. All right, so now we get to the fun part. The movement of electrons through the thylakoid membrane through a series of different protein complexes and electron carriers. So before we get into that, let's take a step back and look at some of what's going in and what's coming out. We already said, and we have our little diagram up here to help us, water goes into the reaction of photosynthesis, oxygen comes out. During that process of photosynthesis, we're also going to produce sugar precursors, and we're going to fix carbon dioxide from the atmosphere. But what we're looking at so far is just the light reactions. So for now, don't worry about the CO2 and don't worry about the carbohydrates. That's part two. Let's just look at the light reactions. And the light reactions, we have light occurring at two places in this diagram, right? This diagram is going to be super important. This would be a great thing to work on in your digital media-free environment. And in the first section where light comes in, there's an electron, and this pink line is following the journey of an electron, okay? As strange as it sounds, this is the journey of a single electron moving through this process, all right? And that single electron starts somewhere, and we'll break this down in multiple ways. So for now, maybe just try and absorb the big picture, and then we'll go into details. The electron has to start from somewhere. So one of the first inputs for photosynthesis is water, right? And water contains hydrogen, oxygen, and because it has covalent bonds, it also has electrons. Well, it has electrons anyway, even though it's separate atoms. But if we break it down into hydrogen ions and oxygen gas, those hydrogen ions no longer have their electron. Remember how simple a hydrogen atom is? It's got that one electron circulating around its little nucleus, right? And so if we were to take that electron away, that nucleus, remember, had one proton. So if we take the electron away, we're left with just one proton. And so that's what these guys are. So where did the electron go? The electron is going to start its journey. That's where the electron comes from, that follows this pink line. So when the electron is here, it's absorbing light energy, and as we talked about, it's becoming photoexcited. That photoexcited electron is going to be passed along to multiple electron carriers. So some of these things aren't doing any work, they're just taking the electron and giving it from point A to point B. Why there's so many, why one couldn't accomplish this, I don't know. I don't know if anyone has an explanation for that. But nonetheless, there are multiple characters here who are passing the electron along, and then it's going to be transported to a molecule that can carry it through the membrane to a second protein complex. So this first thing we're looking at is a complex of many proteins. QB is going to be the transporter from that complex to the next complex. Now look at what the next complex does. This should be very reminiscent of our oxidative phosphorylation process, where electrons power proton pumps to create a gradient. So look at the gradient. Here comes two hydrogens pumped across because of the energy from this traveling electron. There were already two hydrogens from the water. So we are creating an electrochemical gradient, just like we did in aerobic respiration, okay? Now, that electron's not done yet. It's going to be passed to another carrier, and this carrier is going to travel through the membrane. This is, we'll talk about these names, but it's plastocyanin. This carrier is going to donate it to the second complex of proteins. And now, this electron gets a second lease on life. So look what's happening in the second light harvesting complex. Energy from the sun or from whatever light source is bringing that electron energy up again. So this visual representation is it's descending and then ascending purposefully. Okay, so it's showing that the electron now is recharged. And that electron now can do more work. All right, so the second piece of work that it's going to do, it's going to be given to another carrier. So just like PC and just like QB, FD, which is ferredoxin, is going to take it from one protein complex to another. And look at the last thing that's going to happen to this electron. It's not going to go to oxygen and form water like it did in aerobic respiration. At the end of this electron's journey, it's going to be given to an electron carrier, which becomes NADPH, by an enzyme that's called NADP plus reductase. So on the last exam, or the last unit, we talked about oxidation and reduction reactions. When you reduce something, you're adding an electron to it. So this enzyme reductase is adding an electron to NADP plus, sorry. So that electron seems like its journey is done, but what it has created in the process is an electrochemical gradient by pumping protons through and by leaving water to donate those protons. And now that NADPH is going to carry that electron's legacy to the Calvin cycle, which we're not worried about that yet. Now, what happens to the biochemical gradient? I have good news. You already know this. The electrochemical gradient, is going to create ATP through chemiosmosis. Look at this. Hydrogen ions leaving through a membrane protein, producing ATP as they go. That's exactly, if I painted this brown and took off the light and the other stuff, this would be just like what we saw in the intermembrane space of mitochondria. You create a gradient, that gradient does work, ATP is produced. Okay, so this is exactly what we saw before. All right, that's not new material, we're just seeing it in a new place. Okay, and like before, I have a fun diagram to fill in. Can you do that? You all killed the last one. Truly. You had a task, you did it, you succeeded. You're going to be able to do this too. But you're not doing it now, because if you want, and you may not want this, but if you do want, I brought your exams from before the break, and I brought copies of the exams. Bless you, if you're not interested in seeing what you missed, I bid you fond adieu till Thursday. If you do want to see, hang around, and once the chaos in the crowd leaves, I'll try and pass that back to you. So either have a good day or